All right. Well, thank you very much for inviting me. Thank you all for signing up. Like Christine mentioned, if you guys have questions, then go ahead and put them in the chat and she can stop me along the way. I speak a little fast, I know this. So if you ask me questions, I slow down. That's what I tell my students. One of the first things I tell them. So what I want to talk about today is Cytospora canker, and I'll just use some examples from the horticultural trees that we have, trees that we plant. But then most of the talk, I will focus on fruit trees, symptoms, and then some of the research that I'm doing for management techniques and looking at different chemicals that could be applied. So if you have any questions, let me know. This is a picture of a fruit tree, obviously it's peaches, from the Western Slope. And you can see a pretty gnarly canker in the center here. Can you guys see my pointer right now?
Yeah, we can see it.
Okay, good. So this is a pretty gnarly canker that you'll see. So Cytospora canker is caused by a fungus. Sometimes people think that it might be a bacteria, but it's actually fungus that causes this disease. It's been characterized as an opportunistic pathogen. So typically we think of cytosporas as something that prefers weak trees. They can't necessarily invade healthy intact bark, so they actually need some sort of a wound. But that wound could be something like a pruning cut that you might see here, or it could be hail damage, frost crack. So it doesn't necessarily need to be a large wound, but typically they need some sort of crack that occurs in the bark tissue. On peach, Cytospora canker produces a lot of gummosis, and so you can see this response here. This is what we would call gummosis, and sometimes Cytospora canker is called the gummosis canker. That's what some of the literature has mentioned in the past. And then it could also be characterized as leucostoma canker, and that's because the genus, the sexual stage of this pathogen is a leucostoma genus. So if you're looking things up about this, they're all the same thing. Here's another canker that's pretty gnarly that you can see the gummosis coming out of the tree, and here as well. And these are all different cankers. So really interestingly, in the last 5 to 10 years or so, Cytospora, the number of species that have been discovered in Cytospora has increased substantially. Just about every 6 months or 4 months or so, there's another species that's been identified and named because so little is known about Cytospora species. One thing that's really interesting about these is that morphologically, they look really similar. And so people were calling the same species across different hosts. But as we start to understand more and do more molecular technologies like DNA sequencing, we're finding out that the diversity of Cytospora is really great. And so there's very few hosts that don't have some sort of Cytospora that can infect them. So I'm going to highlight the ones that you'd see in Colorado. They might be present on other species, but on other host species, but these are really prevalent and things that you'd see a lot. So the first one I'm going to talk about is Cytospora canker on spruce, and this is caused by Cytospora kuntzii. If you go out into the Front Range near like Rocky Mountain National Park or any forest nearby, this is really common. So up in the Pooter, I see it a lot. Basically what you'll have is a lot of branch cankers that you can see here. And this is sapping occurs, or it's like a gummosis, but it's not yellowish, it's more of a white color, is really common for Cytospora on spruce. And typically what you'll see is that the branches that get hit with Cytospora do often have dieback, and so you'll see flagging that happens across the stem, the tree, across the crown. I don't typically see Cytospora actually hitting the main stem though. A lot of times it's just on these branches that you start to get dieback. But over time, as you get more infections occurring, it can hurt a tree and kill a tree, but not very commonly. Another one. Okay, can you guys hear me now?
There we go. I was gonna say, we lost
Yeah. So another one that we see really often is Cytospora canker on aspen. And it's been associated with poplar borers. So a lot of times if a poplar borer hits a tree, you'll get that sapping, characteristic sapping ooze that you see. And then Cytospora will come in, it'll use that as a wound, and then it'll come in and cause damage. One thing that's really characteristic about Cytospora canker on aspen is you'll get this little outline here. So you can see a yellowing of the bark tissue where the canker, basically the margin of the canker, and which you can see here. So if you see that, typically you have a Cytospora infecting the trees. Cytospora on aspen is so common and it will kill trees. It's a very pathogenic species, or there's multiple species, but it's pathogenic on aspen. And so you will get mortality from this. We think though that more typically you need a wound, and then these trees are a little bit more stressed. There might be drought stress that's impacting them, but it does cause mortality. One thing that's really noticeable too for most of the Cytosporas that I'm going to mention is that you get these fruiting bodies that if you were to run your finger along the branch, it would kind of feel like goosebumps. And so each of these is a fruiting body that then is going to produce spores. And when the cankers get wet and these fruiting bodies get wet, you can get cirri that form. So they're like finger-like projections that sort of unfold, and then they release the spores. So when you see this happening, definitely spores are being released for sure. Another one that's pretty common is Cytospora on green ash. You know, as emerald ash borers come into our state, it's made searching for emerald ash borer a little bit more difficult because you have dieback in the crowns, but sometimes that dieback isn't necessarily caused by the ash borer, it's by EAB, it's actually caused by Cytospora. And so this has been really different than in areas like Michigan and Minnesota where they don't have the level of Cytospora that we have. And so this dieback has really hindered the detection of EAB in certain areas. And so, but it's also very common. And then lastly, Cytospora on riparian species. So a really great example is on alder. Cytospora is known to affect alder trees that are under drought stress. And it's actually been known that it can kill alder trees as well if they're under stress. So those are the ones that we primarily see. And you can see the fruiting bodies here. Here and here. So Cytospora is fairly easy to recognize if you can see fruiting bodies, which they don't always form, and a margin. So if you look up this stem here, you can see a margin where this is a little bit more yellow than here, and that would be where the canker is forming. Some species, it may start out on the branches, but then it also can get onto the main stem. So it can be pretty prevalent and difficult to manage, because once you have Cytospora, and I'll talk about this at the end, the best management tool is basically cutting out the branches that have it. So if it's on the main stem, that tree is likely going to die in the near future. It's a fairly slow disease, but it can still occur. So one thing that's really interesting is that Cytospora species are basically found globally in temperate areas. We have Cytospora that infects pine, maples, walnuts, willows, eucalyptus, elm, and then many fruit and nut trees. This is a map showing fruit trees where Cytospora, the species that we're working with in the Western Slope, Cytospora plurivora, impacts either apples or peaches. And so yeah, in fruit areas, this is a really, really significant disease because once it gets onto that tree, management of that tree is really difficult because it just continues to spread. And I'll show some data that we've collected that looks at how well it sporulates throughout the growing season and throughout the year. And it sporulates year round, which is pretty amazing. And so it can be really important, and they're everywhere, which is important to highlight. Okay, so I'll talk about the disease cycle a bit. You can't have a plant pathology talk without the disease cycle. So basically, the fungus will grow in the bark tissue through open wounds, but some data by Alan Biggs, who was a researcher at West Virginia, show that it can also grow into the buds that you can see here. You can get these lesions that form because it grows on the scales and down. It basically girdles the branches or the tree trunk. So this is a disease that isn't systemic. It's not something like verticillium where it's going to move through the vascular tissue. So where you have a canker, it will just continue to grow and elongate in that region. But any other cankers that you see on the tree are likely a new infection that occurred. It attacks trees when conditions are warm and moist is what we think, but we do know that it can grow year round. We do know for sure that it affects trees that are affected by drought, spring frost, insect damage, sun scald, anything where you had some sort of damage to the tree. These areas are susceptible to Cytospora infection. You can see some sad trees here where you can see flagging. So typically Cytospora will start out in the smaller branches, and then it will move in. Now in peach systems, typically what happens is they prune a tree pretty early on, and they top it. And sometimes even a 2-year-old tree will get infected on that cut wound, and then you've got it in the main stem and it has to grow. But typically an adult tree would start on the edges and then move into the main stem. So the disease cycle is actually fairly simple. You might have a canker that you can see here. The canker then is going to form fruiting bodies, which are those little pimples that I talked about. Those are going to release spores anytime we have humidity or rain. The thought is that this is primarily rain splash or disseminated pathogen. And then if you have leaf scars or damaged buds, it can come in and it'll just start to grow down. And if it's on a branch, it'll continue to grow down until it hits the main stem and it'll continue to elongate. So typically trees may survive 7 years or so with Cytospora if they're older trees, but it will continue to grow. It can be found in the crotches here that you see in in a pruning stub, really common in orchards to see that. Damaged twig, poorly healed wound, and then over time, basically it's going to form these concentric rings. So basically the pathogen grows, the tree tries to respond, the pathogen grows, the tree tries to respond. And so usually these annual rings, if you see them, or annual rings are like growth of a tree. It's basically where the pathogen is infected for that year, and then the tree has tried to respond. And so they're usually, but a Cytospora canker can remain on a tree for many years if it stays pretty small. And then the fungus can persist. This is really important. The fungus can persist on dead wood and on cankers. So if you had a tree that has Cytospora and you cut the branches off that are infected and you lay those under the tree, it becomes a saprophyte and it can continue to sporulate. And so that's one mechanism at which this pathogen can move around. So you'd have basically an inoculum that can spread back into the tree. And so it's really important to remove areas that you've cut off and get them out of a field or in a homeowner's yard or something. So this is a close-up of the fruiting bodies off of peach. So you can see the fruiting body here. And then, so they kind of look like a little pimple, and they'll be pretty common and in high density on a tree that's fruiting, and they can happen pretty quickly. In some of the inoculation studies we've done, after about 2 weeks, we can start to get fruiting bodies that form on a young peach tissue. And then this is the cirri that form. So those are those finger-like projections that basically unfold, and then the spores basically are released. These are the fruiting bodies on an elm tree. And again, I like to imagine touching goosebumps because I think that's really characteristic. So if you see these on a tree, it's typically Cytospora. And what you can do is actually cut these open and you'll see inside pretty organized structures inside. I should have a picture of those, but I don't. But and so they're pretty diagnostic, especially if you cut them and look at them with a hand lens. Any questions? Let me see. Does moisture mean high humidity or soil moisture, or getting tree on the surface, getting wet on the surface of the tree? Either one. So I'll show some data showing just high humidity in the region, but then also if you were to wet the tree, so Cytospora likely moves fairly easily in sprinklered areas, because if you hit like the bark of a tree and while you're watering, then you likely have Cytospora moving around. So it's both for sure. Soil moisture, probably not so much unless that increases the humidity in the air.
I wish I had.
Jane, it looks like Lars has a question. Lars, do you
Okay.
want to unmute and ask your question?
Yeah, hold on. In an orchard, can it go from tree to tree?
Yes, definitely.
So it could damage the whole orchard quite some time.
Right, and I'll show some data that we collected from, we've done surveys in the Western Slope. One thing that the dogma right now is that it's rain splashed. So the movement isn't significant unless you had, what I worry about are those sort of really humid days when they have the fans blowing, because that could move the spores. So any kind of water droplet could move the spores. But typically they're thought of as rain splashed. Does that make sense?
I muted him.
Yeah.
Oh, okay, okay.
And then another question in the chat, if you have sprinklers hitting a tree trunk, is that continuously spreading also?
Yeah, for sure. Yeah, I've been working with growers to get them on drip lines, because sprinklers hitting the tree trunk, if you have a canker there, it would definitely splash and hit other trees for sure. Yeah. And then if you do have Cytospora in an orchard setting, it really depends. So the question is basically, Teresa asked, should we sacrifice the tree or protect the other trees? It depends on where the canker is. So if it's on the branch, you could remove the branch, and then you've sort of protected the other trees, right? So we have a couple of methods that we've developed that I'll talk about later that you could do, but you don't, growers will not sacrifice the trees, because typically if they're newly infected trees, they'll produce peaches for another 3 years. And the peaches might be a little bit smaller depending on how high the infection is, but they're still worth keeping in the ground, right? And so you'll still get spreading of infection unless you either cover the canker where the spores are being released or remove it off a branch. So it gets a little tricky. Eventually the trees have to be taken out, but usually it doesn't happen, economically it doesn't happen until the tree doesn't produce as many peaches, unfortunately. Any others? Okay, so this is some data from Orchard Mesa, where we had basically for an entire growing, or for between October and May, we had cankers where we measured spores once a month. And so what we found is that we can actually, on the x-axis here, we have month from October to May. And then on this axis here, what we have is temperature and relative humidity. So temperature is the black bar, and relative humidity is the checkered bar, the dash bar. And then on the other axis, we have spore counts. So basically what we've been able to connect is that spores are being released whenever the relative humidity is fairly high. So remember, this is Grand Junction, so it's going to be pretty dry. So we're going from relative humidity of about 35 to 60. But you can see here that spores are produced even in December and January when relative humidity is high, which we kind of always assumed that it would stop growing. The Cytospora wouldn't be producing spores throughout the winter season, but it does. So you can get spores that land on a tree. If it's warm enough, the trees are shut down, right? Because they're not photosynthesizing. They can't protect themselves, but yet Cytospora can continue to grow. And then when the trees leaf out again, then they have to respond to the canker that is starting to form with the growth of the pathogen. And so what that really means is that there's really high pathogen pressure, because we can have spores produced in the winter when trees can't respond as quickly. And then they have to respond basically when they start to wake up from winter. Some other results were done. This is in Washington State. And this is by Grove and Biggs. So Alan Biggs was the researcher that I mentioned before from West Virginia University. And he's done a lot of work on Cytospora. He's now retired, but he had done a lot of work. And basically this is 1993, 1994, and 1995. And then months here are in numbers. And then we have the conidia on this lower bar graph in B, but we have precipitation in A. And they found really similar results. So they just found with high rainfall events, they basically saw spores being germinated, or spores being released from cankers. And so we do know that Cytospora does better with some sort of humidity, whether that's rain splash or dew or just high humidity. That tends to be when we see Cytospora reproducing with many fungi.
Jane, we got a quick question.
Yes.
You mentioned like covering a canker. How would you go about covering that?
I'll show you guys, but we have some data to suggest that even latex paint, you basically spray latex paint over a canker. And what that does, it protects, it's a barrier. So you're not going to save the tree that you've sprayed, but it reduces the amount of inoculum that's able then to be disseminated to other trees. And I'll show a picture at the end to give you, it's a pretty gnarly, it's a good picture because it's a pretty gnarly canker that we sprayed. But it does prevent the spread or the movement. And then milliliters, so it's spores per milliliter of water is what we're measuring. So Eric had asked a question. Does that make sense, Eric?
So, but the milliliter of water is collected how?
Oh, so we basically, yeah, sorry. So we basically run water over a canker, put it in a funnel and collect it.
Okay.
Yeah. So we knew exactly how much we were washing onto the canker and then collecting it. And we would do it at the same time every month.
Okay.
Does that make sense?
Yes, that does now.
Okay, sorry. Yeah, I should have described that. Thank you for asking the question. Okay, and then another thing that we wanted to look at was, can Cytospora infect peach at any season? So basically we would inoculate, it was twice a month, and then we just allowed it to grow for about 5 months and then measured. And what you can see is that here is the inoculation date, and then this is the lesion volume that you'd see, and then you can see the distribution. So the cankers are fairly large in the summer months, as you would expect. But even in October, November, December, January, the lesions are smaller because the fungus is basically only growing when temperatures get above freezing, but they're still growing on the tree. So not only do you have spores being produced, but then you can also get infected peaches or any tree likely throughout the winter months, which is really problematic because the tree just can't respond the same way as it would if it wasn't dormant for the season. Okay, and then one other thing that we did was we wanted to look at the canker growth. And so we basically inoculated 200 trees. And then over the next 20 months, we basically measured lesion size, how big the lesions grew. So here's some examples of the lesions that formed over the 20 months. And they're pretty big cankers. So basically what we found is that the average area was about 34 millimeters, and it grew about 2 millimeters per month. So it can grow fairly quickly. And then one thing we know for sure is that Cytospora will grow faster in a stressed tree. So these were at Orchard Mesa, probably, well-watered trees. Peaches are always a little bit stressed because they get pruned typically pretty heavily every year. But in an orchard where there wasn't enough water going on, this would probably be a lot faster growth. So, and I don't know what the response would be in other tree species. It may grow less in, an elm or an ash than it does in peach, but just in general, it's fairly quick spread, I think. Okay, and then the other thing that's really important to start asking is host specificity. So basically, if you have an aspen tree that has Cytospora canker, is your elm tree susceptible to that? And the question is, we don't really know. We know for some species of Cytospora, but some of the data I'm going to show you pretty within the next couple of slides suggests that some of these isolates may have a pretty broad host range. Some of the species might have a pretty broad host range. So this was some work that was done by Jacoby in 2000 and one of his students. And he basically took isolates off of a tree species. So here we have like green ash, and then inoculated those onto aspen ash, which is green, elm, which is orange, alder, which is purple, cottonwood, and willow, and basically asked the question, do we get lesions larger than we might get from just the control, which the control would be the inoculation point, or without any fungus. And basically what he found is that some species like alder, and alder's up here, you can see in the purple, alder really only infected other alder species that you can see as denoted by the star here. So alder really only infected alder. But when we start looking at like green ash isolates from green ash, they were able to infect ash again, but then also elm, which then we're looking at, especially in urban settings where we have tree species of all these different kinds, right? If you have Cytospora on an ash tree, you need to be careful for your elm because it potentially could spread over to that. And then one of the other examples was Siberian elm. It was able to infect, of course, elm, but then also cottonwoods. And so the reason why Jacoby had called these green ash isolates Siberian elm isolates is because the species definitions for these are really tricky. Just, showing at that beginning slide where, every 6 months, 10 more species have been named. We don't really know what the species distribution is, and definitely don't know what the host specificity of each of those are. And so more research needs to be done to understand that, because that's really important for management. If you have an elm, Siberian elm, do you need to watch out for your cottonwood? Probably, because there could be some host specificity or some crossover of those different species. One thing that's really interesting too is some of the research that we've done in the Western Slope shows that the Cytospora that we have on peach is Cytospora plurivora. And when I had first come to the Western Slope, it was assumed that we had 3 different species on peach: Cytospora leucostoma, Cytospora paraleucostoma, and Cytospora cinctum. And then when I started doing molecular analyses, what we basically found is no, we only have 1 species, which was recently named. It was named in 2018, Cytospora plurivora. But as you scatter through the literature, what we found is, even though this has only been named since 2018, it has now been found on apple in the Western Slope. I found it in apple, plum, almond, walnut, olive, and pistachio. So what we don't know is Cytospora plurivora infective to a lot of different hosts, or is it living as a saprophyte? And so that's one thing that we need to start looking at further, but it has been found and isolated from all these different hosts. The other thing that's really important to note is that there are multiple species that are known to infect peach across the United States. One is Cytospora leucostoma, which was the old name that we thought that we had here, but then also a new species that was named Cytospora sorbicula in California and South Carolina. So I'm just really highlighting this out, that it really matters what species we might have, and all this research characterizing these species is super important, but there needs to be more research, because we're not really sure across species what can infect what yet. And there's been little research done on it. I'm hoping to pick that up at CSU. So anyhow, more work needs to be done for that. Okay, so when I first got to Western Colorado in 2015, I was working with Greg Leitis, and he conducted a survey with Harold Larson and of Cytospora in the peach orchards. And what they basically found was that of 200 orchards that they looked at, 100% of the orchards in the Western Slope have Cytospora. This is pretty staggering. And then they also measured how many trees within that orchard has Cytospora. And what they found was that the average of those orchards was 75% of the trees in an orchard in Western Colorado has Cytospora in it. And then they also counted the number of cankers. And what they found was that the average was 5.2. So Cytospora in peach orchards is significant. It's the #1 driver of peach decline in the state. It's not true across the United States. There were other pathogens that affect peach maybe more severely, but because of our weather conditions, I think we get frost cracking that happens because the variation of temperature in the winter. We have pretty low humidity, so we don't get the same foliar or fruit pathogens that you might get in other states. but Cytospora in peaches is a significant issue in the Western Slope, and one that I've been working for the last 5 years to try and help peach growers figure out how to manage this disease. And so what I'm going to do is go through some of the work that I've done. And so this is a comprehensive program for Cytospora management. So I'm going to talk about what chemicals are effective against Cytospora, starting from within the lab to field applications. Then look at cultivars, so are the different cultivars of Cytospora that are equally susceptible, and especially under stress. This is really important because we looked at drought stress and then also pH stress. So the pH in the soils in the Western Slope is pretty high, which may stress the trees. And then also, how is Cytospora moving around in orchards? So the dogma is that Cytospora cankers basically move through rain splash, but we think there might be some other mechanisms. We're working on that now, but I'll kind of talk about that project. Okay, so the first thing we're going to do is look at chemical tests, the chemicals that might be effective. There hadn't been a lot of work. Some people had suggested that Topsin and Captan were effective in benlate, but benlate's no longer an option. And so what we basically did was start with a whole suite of chemicals where we grew Cytospora in plates that are amended with the chemical. So basically, if the plate grew well, like you can see here, or the fungus grew well in the plate that you can see here, it means the chemical wasn't effective. But if you had restrictive growth, then it was more effective. And then we moved from that to the detached branch assays, where we went out to Western Colorado, collected a whole bunch of peach branches, and then dipped the chemicals, which I'll talk about, and looked at which ones were effective against the branches, or against Cytospora on the branches. Whoops. Okay, so here was the list that we calculated. So basically, or that we tested. We had sulfur, we had pristine. This was basically a group chemical controls that growers had suggested to us that we try, and these were our conventional. So we had something like 10 different ones, ranging from multi-site so to specific site action as well. And then we also wanted to try some organic chemicals. So probably about 30% of the growers in Western Slope are organic, 20 to 30%. And so it's really important for us to come up with some mechanisms for them as well to control. Traditionally, neem oil had been suggested to growers to use. And so we wanted to test neem oil for sure, and then some other things. So basically what we have here is we have the chemicals on the x-axis. So here's our control. So this is just growing Cytospora on the plate, as you can see here, without any chemicals amended. It's just the auger. And then this is growth after 5 days. So remember, growth means that the chemical isn't working, and no growth meant the chemical was really effective. So basically, these are our control or conventional chemicals. So we had Alliet, Thompson, Benlate, Captan, and Inspire were effective. And then here are our organic growers. And remember that neem oil was one that they had used previously. It's what they've been kind of suggesting to use. And what we found is that Cytospora actually grows fairly well in neem oil, even though typically for fungi, neem oil is effective, but not for Cytospora. So we basically had NP, Calagreen, Serenade, New Cop, and Badge and lime sulfur. So there were a few chemicals that worked against Cytospora for both conventional and organic. And then what we did was move to this detached assay assay, where we basically we basically took the branches, dipped them in chemicals for 5 minutes, took them out, and then inoculated the branches with Cytospora. So here you can kind of see the wound, and then wrapped that with parafilm to allow the pathogen to grow. And we used all of the chemicals that were effective in the Petri plate assay. And this is what the lesion typically looked like. You can see the wound here. And then this would be the lesion that's formed by Cytospora. So it was pretty obvious when we had a lesion that formed. What we basically found is that it's really interesting, the distribution of chemicals, or the list of chemicals, really narrowed down. So Alliet, even though it worked on the plate, it wasn't effective on the branch. We had Topsin and Captan, which were effective. And then here we basically had New Cop and Lime Sulfur, which we used because they were effective. So this was the positive control, where we basically just put an auger plug on the branch and allowed it to grow. And then this negative control would just be the wound that we formed. We would use a cork borer and remove the bark tissue, and this would just be the size. So in the detached branch assay, Topsin was really effective. I mean, the wounds was the size of the lesions was basically the size of the wound. So then what we wanted to do was take this out to the field. So first what we did was we wounded 1-year-old peach shoots. We then applied chemicals to those wounds. And then we inoculated onto that chemically treated wound that we had done. And this time we used a spore suspension, so it would be more like in the field. You wouldn't have mycelium moving around. You would basically have spores moving around. So we did the assay throughout season. So we did summer, fall, and then spring. And we used Topsin, Captan, and New Cop, lime sulfur. And then here's our positive and negative control. And one thing that's really interesting is that throughout the growing season, the cankers grew at different sizes. So in the summer, we measured canker sizes 2 months post-inoculation. In the fall, we did it at 5 months post-inoculation, and in the spring, then we did it 3 months post-inoculation. But we got similar trends across the growing season, across the year. So this is the chemicals on the x-axis, and this is lesion volume. So again, the smaller the bar, the better the chemical worked. The larger the bar, the longer the bar, means the lesion was able to grow even more. So what we found is that Topsin really is the most effective conventional chemical that we utilized across all 3 seasons. So Captan worked fairly well in the summer, as you can see here, but then that kind of fell off in the fall and in the spring. And then another interesting is lime sulfur was fairly effective, especially when you compare it to the positive control. And it was fairly effective across. It was better than New Cop. One thing that was really important is that New Cop actually had larger lesions than the positive control in the summer season. And we think that the reason why that is because we have phytotoxicity. So it was probably too large of a quantity of copper. And so it caused phytotoxicity to happen on the tree, on that branch. And then we also wanted to use these chemicals, but amended with different latex and lime sulfur combinations. So basically we did the same study, but we used, we amended, so this is latex alone, this is Topsin with latex, and this is Captan with latex. And then we also used Surround, which is basically a calcaline clay. And so it's a material that can be utilized like, latex, but for organic growers. And so basically what we had was Surround alone, Surround with New Cop, and then lime sulfur. And in the summer, what we found 2 months post inoculation was that both latex alone, Captan, and Topsin were really effective. And then for the organics, we found lime sulfur with Surround was effective. But this sort of changed as we went into that fall season and then spring. So 5 months post inoculation, latex no longer is very effective. So basically, if growers wanted to apply latex alone, they would need to do it at least every 2 to 3 months, likely. But then if they amended that with Topsin, then it was still effective. So latex with Topsin was fairly effective. And then if they looked at lime sulfur and Surround, that was fairly effective in both this fall and in the spring. Again, we see the same trend with New Cop, where New Cop, even when it's mixed with Surround, it basically causes phytotoxicity that starts to form. So New Cop wasn't the best situation for growers to utilize. So we have a fact sheet out for this research. So if you're interested in that, you can check this out. And then I'm going to talk about some new chemical trials that we've done so far. So we basically looked at Vitaseal, which is a chemical compound that I'll talk about in a second. But we also did these other treatments where we did Vitaseal with Topsin. We tried 70% latex paint. We were curious of whether or not the cracking was because it wasn't thick enough. And then we also did 70% with Topsin. We tried JMS oil with lime sulfur, and then some other options as well, like new film with lime sulfur. So Vitaseal is a really interesting product. It's a new product on the market. It's a co-polymer emulsion with essential oils. So it's not yet Omni approved, but it potentially could be in the future. It's basically a barrier. So when you have a wounded plant, you could apply this and it would be a sealant on the trees. And it can be applied through painting or sprays, which makes it pretty effective for growers to use. So basically we did the same thing where we cut a branch, applied a chemical, and then we added inoculum. And we did this in the spring of 2018, and then we also did it in the spring of 2019. What we can see here are the different treatments. So thiophanate methyl is Topsin that you can see here, which was really effective in our previous trials. And we saw similar trends for 2018 and 2019, where the Topsin alone was really effective. Vitaseal was effective, but it gets even better if you add Vitaseal with the Topsin. Latex paint again worked both years, but we were checking these cankers at 2 months post inoculation. One thing that did not work very well was oil with lime sulfur. That was another option that organic growers had that they had been utilizing, and it's not very effective. And then new film wasn't also very effective, as you can see here. Any questions so far? No? Okay. Then we were curious about, so we could see, we basically measured lesion size that you can see here, but we were also curious of how these treatments if they were able to actually kill the spores. So what happens if you have no lesion? So once you have a lesion, you know the chemical is not working, but you already basically have establishment of Cytospora. So they might be smaller and they might grow smaller, but eventually you're still going to get a canker that forms. But what if there were no lesions? So basically what we did was we took this material. So we took either a lesioned branch or a non-lesion branch. We plated them all onto an agar plate and then basically tested to see if Cytospora was still alive. And so it really changed what chemicals are effective. So basically what we found is that Topsin is still effective, but then latex paint wasn't at all. So latex paint has basically no properties that are going to kill the spores. So it acts as a really great barrier, but as soon as you get a crack, those spores can actually stay on the exterior of the latex paint, get in, and then cause a lesion. Vitaseal was fairly effective. It doesn't kill the spores 100%, but about 35% viability isn't too bad. And then, of course, some of the organic options weren't that great of a situation, but if you mixed Vitaseal with lime sulfur, this was a great option for growers. And we saw really similar trends for 2019. So one other option for growers is Quava, which we've only tested a little bit. So it's copper, but it's a copper at a lower load. And so we need to test of whether or not it is going to be phytotoxic, but it's Omni approved. And so it might be a great option. So far what we've done, and growers use this typically to treat for coronium blight, which is one other issue that Colorado growers have. It's a foliar pathogen that you can see here that also hits the fruit that you can see here. So we've tested this in plate. So we tested Quava and Vitaseal, and it does appear in the plate to be effective against Cytospora. And we compared it to mustard oil, which wasn't effective at all. But further work needs to be done to take this into the field. So basically what we have now, which growers didn't have necessarily before, was Topsin capped in, potentially Vitaseal, lime sulfur, and maybe Quava. And so there are some options that growers have that they can start applying these. We have always applied them basically on a pruned area, pruned branch. But we need to do some more work to see if we can do more foliar sprays and see how well that is effective. And then these mixtures also work. So latex with Topsin, latex with Captan, Vitaseal with Topsin, and then Vitaseal with lime sulfur. And one thing that's really important to highlight, especially for those of you that are going to be applying chemicals, is that Topsin is a high risk. So it's basically one SNP that changes in the pathogen that then resistance can start to develop. And so what we're asking people to do is basically rotate Topsin and Captan so that you're not using Topsin all the time, because we could get fungicide resistance developing in our populations in the Western Slope. Okay, so are there any questions on the chemical stuff?
I actually have a quick question.
Yeah.
Out of that list of chemicals, like how many of those would be available for homeowners versus growers and maybe certified?
Yeah, probably Quava would be, and maybe Vitaseal. I think those would be the only two. I don't think Topsin is available to homeowners. And then there was one other question is, can insects like the peachtree borer spread this disease? I'm going to talk about that. We're studying that right now to determine if they can. We don't know it yet, but I think I have a hunch that they might be able to. So, any other questions? No? Okay, I'll just keep moving on. So the next question was whether some cultivars are more tolerant to Cytospora under stress than others. So one option is the growers have a lot of different cultivars that they could potentially grow. And in the Western Slope, there's probably 10 to 15 cultivars that are actually grown by growers. So we basically got a collection of these and then wanted to see, how does tolerance vary? Are some more resistant or some more susceptible? So we basically had, I think it was 13 different varieties that growers use. And some of these are from Michigan that you can see here. So they're thought to have a little bit more cold tolerance than these other ones that are from California. So basically what we did was we planted the trees. So we got all these from the nursery, planted them in 15-gallon pots, and then grow them so they would be healthy for about 4 months. And then at 4 months, what we did was we split them into 3 different groups. So for one set, we basically watered them at 100% pot capacity and kept the pH at about 0.7 or 7.0. The second set, we wanted to see water stress. So we basically water stressed them where we watered them only to about 60% pot capacity, but kept the pH at about 7. And then we had a third set where we looked at pH stress, where we watered at 100%, but then we raised the pH by 9. So then after 4 months of this stress treatment, we then inoculated them. And you can see the inoculation point here. And the cankers grow so fast on these young tissues that after 8 days, we basically collected them and measured them. And you can actually see on this this wound here, you can start to see gummosis, and then you can also start to see fruiting bodies. So after 8 days, we're getting gummosis from the tree and then fruiting bodies, which is pretty amazing. So basically what we found here is cultivars on the x-axis and then lesion size on the y-axis. And we basically don't see any significant differences in cultivars, which is, yeah, it's too bad, because it would be really nice to have a set of cultivars where growers could utilize, you know, grow New Havens rather than O'Henrys or something like that. But it looks like there's, yeah, across the board, really no big difference when they're healthy. But then when we start to add the drought stress, what we find is that in all cases, trees that are drought stressed have larger canker sizes. So basically the black bars are where they were watered 100% pot capacity. And then the gray bars are where they were watered at 60% pot capacity. And in some cases though, like in this Cresthaven group, you still get larger lesion sizes, but they're not significantly different. So it may mean that there are some cultivars that can deal with drought stress better than other cultivars. And so that's sort of where we'll head next, is to look at, you know, should Glowing Star and New Havens be grown because they can deal with drought stress, where Cytospora, their lesions are larger, but they're not significantly larger than in others. One thing that's really interesting was this O. Henry, because it was compared in another trial in California, in South Carolina. And basically what they looked at were 4 different, this was Schneibel, who's a professor at Clemson, and they used contender, coronet, O. Henry, and summer prints, and basically challenged it against Cytospora. And what they found was that O. Henry was one of the best cultivars to grow against Cytospora compared to these other varieties, which aren't grown necessarily in Colorado because the Southeast grows different cultivars than we do. But it was really interesting. Another thing too is that they looked at 2 different other canker pathogens, Botrytis and Phomopsis. And what they find is Cytospora, unfortunately, is the better grower on peach trees. So then we also wanted to look at the pH. So treatment, so basically the black bars are pH 7, and then the gray bars are pH 9. And what we found is that there's little effect of pH, which is great news for the growers because it's really difficult to manage pH stress in the field. So they can use these sulfur burners, which they sometimes use, but it doesn't necessarily look like peach trees are responding to that type of stress. To have that higher pH doesn't necessarily cause larger lesions to form. It does for some cultivars, but not others, and none of them were significantly different. There was a lot of variation across those treatment groups. So what we've done now is we planted the trees out at Rogers Mesa, and we're going to continue this trial, but actually in the orchard. So these trees have been planted for 2 years now. And so we'll do inoculations in the middle of the month and evaluate if they're in the field, do we still see that difference? And then we're going to do the drought treatment, but not necessarily the high pH. So we'll see how, if we see cultivar difference in drought stress trees at that higher pH as we see in the orchards at Rogers Mesa. Any questions for the cultivar susceptibility before I move on to how's Cytospora moving around?
We have one question back to like the chemical treatments.
Yeah.
What would be, if you were to mix up the topsin plus latex, what would the ratio be?
It's usually 50-50. Or it's like, no, 3% topsin. So it would be 50, because you would mix 3% in water, right? For the, for the topsin, and then 50% latex. Does that make sense? How I described that?
Yeah, so you make your 3% mix, and then you make your 70% mix, and then you mix them together?
Yes, exactly. Yeah.
50-50. Okay.
And I think the treatment for 50% latex with topsin was fairly similar. So I wouldn't necessarily recommend 70% latex. I would recommend the 50%. Because we didn't really see too much of a difference.
Perfect. Hopefully that helps, Sydney.
Okay, so one thing that we're really curious about is how Cytospora is moving around. So this is some work by Grove and Biggs, and what they showed was basically that the maximum spread that they get from rain splash is about 2 feet. So here you can have the horizontal distance from a canker, and then this was the number of colonies. So basically what they did was they set out plates in front of a canker in different angles. So they did 0, 90, 180, and 170 around a canker. And then they looked at how many spores basically dropped onto that plate and measured them. And what they found was that it was basically about 2 feet, and you had more spores at higher angles than 90 degrees. So there's not a lot of spread, you know, from a canker, from rain splash of a canker, which is kind of amazing. But when you go out to the orchards and look, what we would expect is if there was really just rain splash, you would expect to see these small foci of infected trees. So imagine this is an orchard. The green ones are healthy. The green dots represent healthy trees, and the red dots represent sick trees. You would expect just these little pockets of spread to occur. But what we see basically is these, you know, random trees. You might have a lone tree in one area that's got Cytospora, and then you might see these same pockets, but it's really dispersed differently than what I would have imagined. So basically the question is, what ways are spores, how do they travel long distances? Is it through wind, insects, or humans? So what we've done so far, and this work isn't completely finished yet, but what we've done is we developed a digital droplet molecular assay. So it's basically where we can take a branch tissue or we can take an insect and then molecularly detect whether or not we have Cytospora florivore on the samples. So we collected aerial samples, insect samples, and then we also tested nursery stock. So one thing I'm wondering is if it's actually moving in on nursery stock into the orchards, because that could be another mechanism for spread. So last summer, we collected air samples and insect samples from 6 orchards. So we had 3 conventional growers that we worked with and then 3 organic. And we set them up basically in this X-like fashion across the orchard. And then we collected peach twig borer, metallic wood borer, and the greater peach tree borer. And then once every 2 weeks, my graduate student, Stephen Miller, basically used a filtration system. So basically this is a pump that filters air into a plate right here. So this is a Petri plate inside. And then measured, basically looked at how many spores were on that plate. And over the summer, this is the distribution of what we collected. So this is from June to August, and this is the number of insects we collected. Blue is the peach twig borer. Orange is the greater greater peach tree borer, and then red is the metallic wood borer. And basically, what we found was about 2,800 different insects that my student is still going through right now. So basically, the insects were placed in vials, and then they'll be rinsed, and then we'll collect all the water from those. The aerial samples were rinsed, and then the spores were placed in water. And so right now, he was supposed to have everything done this spring, but because of the situation with COVID, that's been kind of on hold. But the samples are currently being tested slowly but surely. And then we also got about 3,000 peach trees from 4 different nurseries this year, which we half of them we basically sampled immediately after the truck. So we sampled from the branch tissues and the root tissues and then the main stem to see if there's Cytospora on the trees. And then we also have half of those trees that we're gonna stress through drought stress and see if they form Cytospora cankers on them. One idea is that Cytospora is an endophyte, so it's basically living in the tree but not causing disease until the tree starts to get stressed, and then you get symptomology occurring. One thing that's really cool is that with the method, the molecular tool that we've developed, we can basically detect Cytospora after only 4 or 5 spores. So these blue dots indicate that Cytospora is in a sample, and that was after we diluted it so much that we only had 5 spores per mil, which is really exciting. So if it's being vectored by either the nursery stock, the wind, or insects, we should be able to detect it with this molecular technique. And hopefully we'll have results fairly soon on that, which is really exciting. Okay, so I talked a lot about peaches, but I kind of want to go back now to how do you manage Cytospora. This is a distribution of hosts for Cytospora that came out of this paper that just came out in June 2020. And basically what we have here are 6 different species or 5 different species. You can see that Cytospora looks fairly similar across the different wood types. Now these are pretty young trees, but you can still see the fruiting bodies, and you can still see the cirri that are forming here and here and here. So Cytospora, I think, if you have fruiting bodies, it's a disease that you can diagnose fairly well. One thing you want to make sure of is that you don't think of sapping as a symptom on all trees. So for some trees like alder, you won't get any sap. You'll basically just get a sunken canker. You'll just get a sunken canker, but you won't necessarily get the sapping. Here on the Allianthus, you can see that there was a lot of sapping that occurred. And gammosis can happen if a tree is damaged, but we on some tree species, we do see it a lot associated with Cytospora. There was a question by Teresa. If a homeowner has gammosis on a tree, is Cytospora the likely culprit, or are there many other causes of gammosis? Is there any way to know for sure it's Cytospora? Gummosis is a general term. Sometimes you'll get it with insects, but I don't see it that often without Cytospora, but it doesn't always have to be Cytospora. If you see fruiting bodies surrounded by the gummosis, then you know for sure you have Cytospora. If you just see sort of a canker with the gummosis, it's not a sure thing, and so you probably want to get it sampled from the clinic, the CSU diagnostic clinic. But the fruiting body should be present, and you can see they are typically very prolific, and if you see that, you probably have Cytospora. The chances are very good that you have Cytospora. Okay, so how can you manage Cytospora? We know this is a pathogen that doesn't like healthy trees. It likes stress trees. So one is proper planting. You want to think about future stress, prepare the site, plant it well. If it's got any sort of girdling or anything, it's going to be stressed. It's not going to get the same water that it would. That could cause Cytospora in the future. Management of water stress. This is super important, especially with homeowners. You want to make sure they're not just watering their grass, but they're watering for trees. We don't know much about nutrient stress and Cytospora. That could impact. We'd need to do some more research on that. Fertilizing in the winter and in the summer, but you want to not fertilize too late in the fall. And then prune correctly, and I'll talk about that in a second. I only have like 3 slides left, so this should be fast. You want to slow the growth in the fall. So especially in Colorado, where we've got these really variable winters where you can have drops of like 50 degrees, the trees need to be hardened. So if you add too much fertilizer too late in the season in the fall, you'll get newer growth. And then any damage that happens, even if it's on the twigs, can basically, Cytospora can start to impact. And really, we know that a common source of damage is cold injury in peaches. And so you can see the cankers that have formed because of frost cracking and stuff. And it's hard to manage, but you can at least prepare the trees a little bit. One thing about pruning, so we know that poor pruning can establish Cytospora. So one thing that we suggest is pruning often. So you want to be able to make smaller cuts rather than larger cuts. So if you wait to prune a tree and you've got a really large branch that you didn't have to cut off, you basically then have a larger surface area for Cytospora spores to get in there and start to cause infection. You definitely want to cut out the cankers on the branches. If you've got cankers that you see, you want to prune them out, and remember to remove them off site. It's not a good practice to keep them on site because Cytospora basically just turns into a saprophyte, and it can continue to sporulate and then cause spores to come up. It's been suggested that you could describe cankers on the stem, and I know some peach growers do that. So if you had a canker that was forming like here, you could scribe around it, and basically the tree will then form wound tissue, but you don't necessarily, this will dry out, and you wouldn't necessarily have Cytospora that continues to grow. The only issue is that, like in this region here, you might have fungus growing beyond where the canker is. And so you can scribe it, but you may not have gotten all of it. It may slow the growth of the canker, but I don't recommend doing that on many trees, especially large trees. And then Biggs in 1989 looked at different pruning techniques. So we basically compared the stub with the collar with the flush. And then after those pruning cuts, looked at how many were naturally infected with Cytospora. So here we have the pruning technique, and then we have visible necrosis, leucostoma, Cytospora isolation. So remember, this is the sexual state of the fungus. How many produced gumming, and then how much had a callus tissue. And basically what he found was the collar was the best method, because he did see necrosis occurring about the same as in the flush, but he wasn't able to isolate Cytospora as readily as with the other two. So this type of a cut is a much better prune cut than either the stub or the flush. And so that's important to recognize. For the reducing inoculum, again, pruning infected branches is super important. Clean your prune tools. You don't want to move it around from one area to the other. You could use ethanol, you could use alcohol, Lysol, Clorox, anything would be great. Some growers are starting to use chemicals that they apply to the pruned branches. Typically they will either use latex or they'll use latex and Topsin, but it's nice to be able to see the area that you've basically controlled. And so that can be really important. And then also, this is our treatment of a spore. So this is a pretty gnarly canker that you can see up here, or treatment of cankers. And then what we've done here is basically applied latex over the canker. And my graduate student basically did the same thing that we did when we were measuring spore concentrations across the growing season, where he ran water through for an entire year across this painted area. And there's a pretty good reduction for at least 3 months for the latex alone, and then at least 5 months when you amend it with topsin. So basically you're not saving that tree, but you are reducing the amount of spread that you'll see across a site, which can be really important. Okay, that's all I have. So thank you so much for inviting me. I work with a lot of collaborators. We have the Cytospora Working Group, which is a group of growers, extension personnel, CSU researchers, and we get together about twice a year and talk about some management techniques, some research questions that we need to do. So we really try to incorporate the community in the research we're doing, because they're driving the research that we've done. We work with CropWorks, Giannis Minas and David Sturley. Giannis is a professor at CSU in pomology, and then David Sturley is his technician. And then we also have worked with Meredith Schrader for collecting the insects and stuff. And so there's a lot of great people that we are working with across the Western Slope and beyond for this project. Okay, do you guys have any questions? And if you have anything that you want to talk about, send me an email. My email's up there. If you have any questions, let me know. Sorry, that was a little long.
No, that was perfect. Thanks, Jane, so much. Yeah, if people want to type their questions into the chat, or if they want to do the little fancy hand raise situation, that's pretty fun. Like I said, I scheduled us till 3:30, so we got some extra time. And I guess I'll also point out, Katie has put into the chat a link to our evaluation form. So if you would so kindly please fill that out either right after this, or if you want to sit and ruminate on it for a little bit and then fill it out, that'd be great too.
I'd like to thank you. It's very informative. This is
good.
Thank you.
Thank you.
Lars, did you have another question?
Yeah, just so as I'm driving through Fruta sometimes and I see a bunch of peach trees or something, or apple trees. Some of them are painted up from the bottom up, like a white paint. Is that the latex that you're talking about, or is that something different?
No, it's typically latex that they paint with, and it's typically for sun scald. So it's basically to protect against bark cracks that happen lower down. Yeah, but it's not necessarily a Cytospora treatment, but it is to manage for abiotic damage.
Okay, thank you.
Yep.
And it was very good.
Oh,
very
thanks.
good.
My name's Cindy.
Hi,
Hello.
Cindy. Yep.
Great presentation, really good.
Thank you.
We learned a lot. We have a little orchard.
Oh, good.
The only suggestion I could make is maybe have the arrow pointer a little bit bigger
Okay.
when you're zooming around, because I could hardly
Keep up
follow
with it.
it. But I you had very good explanations for everything. And
Oh, good.
it was great. Thank you.
Thank you.
Well, maybe you've blown them all away.
It was nice to have questions during the middle.
Absolutely. Absolutely. Yeah, so I don't know if you want to remind people of your email address again.
Yeah, it's jane.stewart, Stewart, @colostate.edu. So if you have any questions about, Cytospora or other pathogens, let me know on trees.
Perfect. Well, I'll go ahead and stop the recording.